Good morning, students. I hope that you all are following all the topics that we have studied under microbiology. Today we are going to study about microbial enzymes. Nowadays, instead of chemical catalyst, microbial enzymes are used in chemical industries. We know that a catalyst speeds up a chemical reaction. It increases the rate of a chemical reaction. So chemical catalysts are used on a large scale for mass production of various substances, various other chemicals. But nowadays, instead of chemical catalyst, microbial enzymes are used. Now we know what are enzymes. Enzymes are biological substances or macromolecules that are produced by the living organism, and they act as a catalyst to bring about a specific biochemical reaction. So enzymes that are produced in our body are used as catalysts to speed up the various biochemical reactions. But here we are talking about microbial enzymes. To just give you an example of a microbial enzyme, we have studied about vegetarian cheese being produced nowadays. Earlier we know that it was obtained from rennet that was obtained from the cattle, the alimentary canal of cattle. But nowadays we are using an enzyme protease that is obtained from a fungus to make cheese. So protease is a microbial enzyme. So why these microbial enzymes are gaining popularity instead of chemical catalyst is what we are going to study today. So just let us see what are the characteristics of these microbial enzymes. Active at low temperature, pH, pressure. So these microbial enzymes, they are active at low temperature, that is they will work at low temperatures, pH as well as pressure. They do not require very high temperature to get activated and catalyze the reactions. So they are active at low temperature, pH and pressure, and that is why they are used in chemical industries. Saves energy. So if they are going to be active at low temperature, pressure and pH, obviously they will be saving energy. Erosion proof instruments are also not required. Many chemical catalysts require erosion proof instruments, but this is not the case with microbial enzymes. So erosion proof instruments are not required. Unnecessary byproducts are not formed, and hence expenses on purification are minimized. Many chemical catalysts give rise to unwanted products, and hence the product that you want needs to be purified. You need to remove all the unwanted products, and hence you have to carry out purification processes. But the case is not with microbial enzymes. They do not produce unnecessary byproducts, and hence time, money, all is saved due to this. Enzymes can be reused and hence are eco-friendly. So enzymes are eco-friendly because they can be reused again and again. You get the same quantity back, and the microbial enzyme can be used again for the process. Now, what are the various types of microbial enzymes? So, according to the International Union of Biochemists, enzymes are divided into 6 functional classes, and they are classified on the basis of the type of reaction which they are used to catalyze. So these are oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. So 6 different kinds of enzymes are there. And the classification is done on the basis of the reaction they catalyze. So just to give you a brief information, oxidoreductases enzymes are the ones that catalyze the transfer of electrons from one molecule, that is the oxidant, to another molecule, that is the reductant. Hence they are called as oxidoreductases. Next is transferases. Transferases are enzymes that catalyze the transfer of group of atoms, such as amine group or carboxyl group, etc., from the donor to the acceptor. So they are transferases. They are used for transferring a group of atom from the donor to the acceptor. The next one is hydrolases. Hydrolases is a class of enzyme that performs the function as a biochemical catalyst, and it is using water to break a chemical bond. Since water is used to break a chemical bond, it is called as a hydrolase enzyme. After that we have lyases. Now this lyase enzyme is used for catalyzing or breaking of various chemical bonds. So lyase enzyme is used for breaking chemical bonds. Next is isomerases. Now isomerases is the name given to the class of enzymes which are able to convert molecules from one isomer to another. Now isomers we will study in carbon chemistry, but just to give you a brief idea, isomers are those substances which are going to have the same molecular formula but different structures. So compounds that are going to have same molecular formula but different structures are called isomers of each another. So here we are talking about the isomerase enzyme, and it is the one which is going to bring about the conversion of molecules from one isomer to another. The next is ligases. Now ligases are enzymes that are capable of catalyzing the reaction of joining two large molecules by forming a new chemical bond. So the basic difference between lyases and ligases is that lyases breaks chemical bond in order to form new compounds, whereas ligases forms chemical bonds in order to combine different compounds. So this is what is the basic difference between the two, that is lyases and ligases. So depending upon the functions that they perform, they have got different names. So this is all about the microbial enzymes. Applications of microbial enzymes. Easy removal of dirt from clothes at low temperature as detergents are mixed with enzymes. So by making use of detergents that are mixed with microbial enzymes, we can remove the dirt from clothes quickly. Proteases, lipases, amylases are the major class of detergent enzymes. So all these three types, that is proteases, lipases and amylases are used in detergents. And each of these provides specific benefits for easy removal of dirt from clothes. And it is even used in automatic dishwashing. Stains generally are removed by mechanical action, but this action is enhanced and the removal of dirt from clothes takes place quickly if the detergents are mixed with microbial enzymes. Proteases are the most widely used enzymes in detergents, and various kinds of stains can be easily removed. Glucose and fructose syrup is obtained from corn flour by adding enzymes obtained from Bacilli streptomyces. And the third application is microbial enzymes are also used in preparation of cheese, plant extracts, textile, leather, and paper. So these are some of the uses of microbial enzymes.